Good evening, everybody. Uh, I welcome you all again in our online lecture series. Now, in today's class, I'll mainly concentrate in the DNA restriction enzyme, or not the restriction enzyme, but DNA modifying enzyme, or the cloning modifying enzyme. Now, the DNA modifying enzyme, I can classify it in different types. Number one, endonuclease. Number two, exonuclease. And mainly the two types in the endonuclease and exonuclease. But within the endonuclease, we have already studied about the restriction endonuclease. Apart from the restriction endonuclease, there are other two types of endonuclease. One is DNase 1 and another one is S1 nuclease. And within the exonuclease, BAL31 and exonuclease 3 we can use for the DNA modification. So DNase1, what is the function of DNase1? It can chop the double-stranded DNA in the intercalary or the intermediate region at random sites. That means the DNA will be fragmented and we will get the DNA in the multiple fragments. And as well as the S1 nuclease also chop the DNA, mainly the single-stranded DNA at random sites, and also we can get the fragmented nucleotides with the help of the S1 nuclease. Now, the exonuclease activity is mainly here driven by BAL31, and this BAL31, it cleaves the DNA from both sides, from both end, 5' end and as well as the 3' end. So with the help of the exonuclease or mainly the BAL31, we can make a blunt-end product. And with the help of the exonuclease 3, the exonuclease 3 shows the exonuclease 3 shows the activity, exonuclease activity from 3' end. Exonuclease activity is mainly found from the 3' end in case of the exonuclease 3. And so with the help of the exonuclease 3, we can leave 5' end overhang to the amplified product, and these are the digested product, and the digested product just resemble the staggered end product. So with the help of the exonuclease 3, we can make the blunt end product to staggered end. Now, apart from the endonuclease, apart from the exonuclease, there is another ribonuclease. Ribonuclease, we know that it can cleave mainly the RNA. That is nothing but the RNAs. Now, the polymerase, different types of polymerase are also used for the DNA modification. Number one, DNA polymerase 1. Number two, clenofragment. And number three, reverse transcriptase. Now, DNA polymerase 1 and the clenopolymerase has the property to synthesize the DNA in the 5' to 3' direction. And DNA polymerase 1, it has also the property of the exonuclease activity, but the clenofragment is the modified version of the DNA polymerase 1 and which doesn't possess any exonuclease activity. It should possess only the polymerase activity. And this clenopolymerase is mainly used when it will be used, it is used mainly to produce the blunt end product from the staggered end. So staggered end product can be made blunt end with the help of the clenofragment or clenopolymerase, and as well as during radio labeling, during southern blot, we are using the radio level probe, and during this radio labeling, we can use this clenopolymerase to incorporate the radio level nucleotide. Now, there are two examples where we can use the exonuclease 3 or the clenoenzyme to make a blunt end product from the sticky end. Here in the first, this is the overhang, which 5' overhang sticky, and this is the product from eco R1 digestion. So eco R1 digestion, and then how can I make this eco R1 digested product to the blunt end with the help of the exonuclease or the cleno? Here we should use the plano, because 5' to 3' polymerase activity should be there, and that's why with the help of the plano enzyme, I can produce, or we can produce the plant-end product. Now, for the second example, there is the 3' overhang sticky end. The staggered card, definitely, but there is a 3' end overhang. So exonuclease 3 or the T4 DNA polymerase, it shows the 3' exonuclease activity. So 3' end overhang should be removed by exonuclease 3 activity, and thus we can make this product blunted. So with the help of these two different enzymes, we can produce the sticky end product to blunt it. Now, other end modifying or DNA modifying enzymes, depending on the phosphorylation, dephosphorylation. First one will be the alkaline phosphatase. Why alkaline phosphatase? Alkaline phosphatase shows the phosphatase activity. Phosphatase, it will remove the phosphate group from the 5' end of the DNA or the amplified product. So 5' end of the phosphate, when will be removed, it leaves the 5' OH group. And in both the cases, if we have the 5' OH and 3' OH, then it will be never ligated. So this advantage of the alkaline phosphate, or this use of the alkaline phosphatase, is commonly used where to avoid the self-ligation of a vector. If I choose only one type of enzyme for restriction digestion for vector, and then how can I restrict, or how can I prevent the unwanted ligation or the self-ligation? Because if we use only the single enzyme, then always there is a chance for the self-ligation, mainly for the vector. So how can we prevent it? After digestion with the single enzyme, I can use the alkaline phosphatase. Alkaline phosphatase will then and there remove the 5' end phosphate and make it the 5' end OH. So once the 5' OH and the vector leaves the OH in the 3' end and as well as the 5' end, so it will never ligate. So in the example, in the cartoon, it is quite clear that the vector having 3' OH and the vector having 5' OH, after processing with the alkaline phosphatase, it will never ligate it. So self-ligation can be prevented. Now, the digested vector will be treated with the alkaline phosphatase. So 5' end OH, no phosphate, and 5' phosphate is only present in the insert, because the insert is not treated with the alkaline phosphatase. Insert is kept as it is, and the vector is only treated with the alkaline phosphatase. So vector in the 5' end there is OH, and in the insert there is a 5' end phosphate, and in the 3' end of the vector have OH. So the ligated product will be the recombinant vector. Only insert can ligate it with the vector, the digested vector and the alkaline phosphatase treated vector. So we will get only the ligated product of vector and insert. So we can check, we can restrict the self-ligation of the vector. Now, second group is the terminal transferase. Here we can add the homopolymer or the nucleotide at the 3' terminal. Terminal transferase means the transferase activity of the enzyme will be shown at the 3' terminus or 3' end of the vector. Now, when we will use, suppose if we don't want to use the restriction enzyme, just directly, if we clone our cDNA or the PCR product or any amplified product directly into the plasmid, how can we use it? There should be a linearized plasmid, and in other hand, there will be the amplified product. Now, this amplified product and the linearized plasmid will be treated with terminal transferase along with the nucleotides. That polymeric nucleotide, homopolymer, the multiple nucleotides, and the nucleotides maybe DGTP, maybe GGGGG, maybe CCCCC, or A, poly C, poly G, poly T, whatever it may be. So if I can add DGTP, poly G will be added to the 3' end of the vector in the both termini, 3' and 3' end of the both strand. If we add poly G, poly C, or poly T, then poly T will be added in the 3' end of the both the stranded of the vector. Now, if we add the poly G, then we should choose the poly C for adding into the cDNA or the amplified product or my gene of interest. So poly C, if I add the poly G for the vector, poly C will be added in the both the end, both the 3' end of the both strand of the cDNA or the my gene of interest. Then, depending on the complementarity, this poly C product, poly C carrying the gene of interest, carrying this poly C, will bind with the poly G treated or the linearized plasmid carrying poly G. Then this nick is sealed with DNA ligase. So ultimately, without using any restriction enzyme, we can easily get our recombinant product. So terminal transferase is very useful. Topo TA or TA vector, TA cloning is one of the examples of this type of activity that depending on the complementarity, insert is easily ligated to the vector depending on this TA complementarity. So this is these terminal transferase is commonly used for cloning purpose, and there are other two. There are also other two sites that can be used, also used for the addition at the 3' end, like adapter and the linker. Within the adapter, we can choose the different restriction site, and this restriction site will be present within the adapter region, and this adapter region can bind with the cDNA or my amplified product, and then I can use the restriction enzyme, and with the help of the restriction enzyme, I can make this cDNA as the digested product. So these are the several examples we can use. We can modify according to our need in the DNA cloning or the gene cloning for the gene cloning purpose. And next, the polynucleotide kinase. Polynucleotide kinase means the phosphorylation activity. So it can add the phosphate to the 5' OH. So the 5', that means the digested product will be phosphorylated, and that can be used for further ligation. And this PNK, all polynucleotide kinase, is also used, frequently used in radio labeling. When in the southern blot, in the polynucleotide kinase is very commonly used for to prepare the radio level probe. So that's all about DNA modifying enzymes. In the next day, in the next class, we will concentrate on the different types of vectors, the cloning vectors. Thank you all.